So our last speaker of this session is Nuno Brito Santos from the CRIC, and he's going to tell us all about the RDRP and its association with the viral genome.
Hi, my name is Nuno Santos. I'm a postdoc at the RNA Virus Application Lab at the CRIC, and I'm going to talk about the dynamics of SARS-CoV-2 polymerase association with the viral genome. Coronaviruses are positive-sense RNA viruses. They have a large genome, and therefore they need an RNA-dependent RNA polymerase to replicate their genomes, which is an sp12. I'm sure you're all familiar with this kind of genome representation, but in reality it looks a bit more like this, so it's highly structured, and these structures are probably very dynamic, which poses quite a big challenge for the polymerase to replicate it. So our kind of broad question is, how does the polymerase deal with all of this complexity? So the first thing is to understand if we can find an sp12 associated more frequently with specific genomic positions. Then we want to integrate this with what is known about the mutational fitness and the structures, and then we can generate educated guesses about biological function. So the technique that I've been using is cross-linking and immunoprecipitation, CLIP-seq. So very briefly, we infect VIR-6 cells with SARS-CoV-2 at a high MOI. Then we UV cross-link protein to RNA. We immunoprecipitate our protein of interest, which is an sp12. Then we ligate an IR adapter that allows us to run the samples in a gel, transfer to a membrane, and actually image them. So according to the molecular weight of the protein plus RNA, we can decide where we extract them from the membrane, purify. Then we digest the protein, which leaves just a tiny peptide at the cross-linking site. So when we perform the reverse transcription, the reads will stop at the cross-linking site. So finally, we do the library prep, we sequence, and we can do the peak calling by analyzing where the reads stop. So this is the kind of data that we get. So if we have lots of reads, ending in a specific position, this tells us that an sp12 was bound there at an individual nucleotide resolution. So then we can make this a bit more user-friendly and convert this to this kind of plot where we have in the x-axis the genomic position and in the y-axis the number of cross-links. Also, to be sure that this is really not just randomly reads ending there, we have a control that was treated exactly the same way, but there was no UV crosslink in the beginning. So here we'd expect just to have like random RNA being carried over. So an interesting result would be something that has a peak in the experimental condition that is absent in the no UV. So this is what the real data looks like. It's not surprising that we can see an sp12 all over the genome because polymerase is interacting with RNA. But we can see that there is like a big peak that I'm going to address today. Are for future work.
A little bit closer.
Oh, sorry. We have this amazing peak, but unfortunately it was just an artifact. But if we focus on the 3' UTR, we have this massive peak. So let's focus on this one. So in coronavirus, the 3' general organization is kind of conserved. So we have the bulge stem loop, we have the hypervariable region that, as the name indicates, it varies a lot, even though it contains the highly conserved octanucleotide sequence. And we have this loop that can form a pseudoknot with the basis of the bulge stem loop. So if we zoom in the 3' UTR in our sp12 clip data, we can see that we have these peaks here, and they actually map to the bulge stem loop. We also have further signal in the hypervariable region, and this was at 6 hours post-infection. If we do the same analysis 10 hours post-infection, we can see that these peaks get even more pronounced. So we are quite sure that an sp12 is binding the bulge stem loop. And why is this relevant? So this bulge stem loop has been proposed to be a molecular switch involved in the initiation of transcription. So what has been proposed is that when the switch is off, the 3' end is paired with the L1 region, and there is no pseudoknot. Then the 3' end is released from the L1 region. The pseudoknot forms, and this allows transcription to initiate. So obviously for this we need an sp12, and I've showed you that an sp12 is here, but we also need other accessory proteins, so an sp7, 8, and 9. So this is quite preliminary, so take this with a huge pinch of salt. But you can see that an sp7 that forms a complex with an sp8, and it's involved in stabilizing the polymerase complex. We can see an sp7 signal upstream of the bulge stem loop, a bit in the bulge stem loop, and also throughout the hypervariable region. an SP8, which is a primase in vitro, and it interacts directly with the L1 region. We can also see that it's in the hyperverbal region and also a bit of signal in the L1 region, which is reassuring. And finally, an SP9 that's involved in capping and binds both ends of the genome during replication. And yeah, this is being very optimistic, but it seems that we can also find an SP9 in this region. So taking all of this together, what we think that is happening is that we have the polymerase complex sitting in the bulge stem loop, and then towards the formation or not of the pseudoknots, this will make the 3' end of the genome interact with the base of the bulge stem loop and recruit the polymerase complex to initiate transcription. This is also interesting because there is a resolved structure that has a double-stranded RNA that is exactly the size of the bulge stem loop. So to finalize, I've showed you that an SP12 binds the bulge stem loop, and we are quite sure of this. I've also showed you, not so convincingly, that an SP7, 8, and 9 can be found there. And this is because I've tried this again, but it's not super reproducible, probably due to technical limitations of the antibodies. So right now I'm optimizing new nanobodies to improve the IP efficiency and therefore having more data. Also, I've showed you that an SP12 binds the bulge stem loop and the end of the genome, but to be sure that it's really an interaction between the 3' ends and the bulge stem loop, it would be good to see the same NSP12 molecule interacting with both RNAs. So for that, I'm also trying to do high clip, which is another technique that will allow us to detect protein bound to RNA that is bound to other RNA. We are also interested in interactions between the 5' and the 3' end because they happen during replication, even though it's not totally understood how. We've also been working with some molecular dynamic simulations to try to understand if the formation or not of the pseudoknot, if it makes it more or less likely for the 3' ends to interact with the bulge stem loop. And the next thing will be to do reverse genetics and mutate an SP12 binding sites and see what happens to the virus. Finally, we've been doing a collaboration and try to understand if this 3' end structure, including the bulge stem loop, is sufficient to initiate transcription in vitro. Now I want to end by thanking my supervisor, David Bauer, my colleagues, also all of our collaborators, the funding agencies, and thank you for listening, and I'm happy to take any questions.
That was a really cool talk. Questions? There.
Thank you. Very, very cool talk. A question that came to mind was like, if you tried to do it like at different time points, because like, for example, during viral replication dynamics, we know that at the beginning you get a lot of subgenomic mRNAs for N, and then you get less and less subgenomic mRNAs of everything else. And maybe this is not relevant, but if you, like, would you be expecting to see NSP12 in the cross-linking in these clips at other parts of the genome if you do it at different time points?
Yeah, I've tried 6 hours and 10 hours post-infection already. Yeah, it would be great to do more, but the issue is that this is quite a lot of work.
No. Fair.
So, yeah.
Andrew.
Yeah. I just wondered, do any of your peaks correlate with sort of TRS bodies? Like you would expect maybe the polymerase pause at certain points?
Yes, especially in the 5' UTR, we can see that. But what we have been analyzing recently is if you are detecting subgenomic RNAs or the genomic RNA. So it's still an ongoing analysis. But yeah, we can definitely detect it in the TRSs.
Okay, if that's it, I think we all deserve a coffee. And back here at 11:30. Thank you very much, and thank you to all our speakers. Thank you.